necessary. Well, here we are. So this is physics and chemistry are all about artificial intelligence. I mean, yes, there are names there, but it's about AI. And you may remember that I did a series on AI this time last year, so that plays right in these two this year. Last week was interesting, hopefully this even more so. I'm certainly very keen about this one more than last week, and I like last week too. That's just an early morning sketch. I think it took me kind of 10 or 15 minutes, but it just shows you why, and an easterly wind. The wind is coming this way, so that's why it's kind of thing. So it's kind of pretty. But, you know, there's a lot of beauty in nature. And this is, we're talking about proteins here. This is a very sophisticated protein. This is the largest molecule in the body. There are hundreds of these pores in the nucleus, the nuclear membrane of a single cell. Can you imagine the complexity of this? And this is a pore that opens and closes. For example, it allows messenger RNA to get out from the nucleus out into the cytoplasm where it can make proteins. So, so it's a wonderful world, but like a lot of beauty in nature, if you can't see it, you can't appreciate it, or you don't know about it. So this is about knowing about at least some of it. And moving on, well, it wouldn't be the Nobel Prize series without some comments on it. You know, the latent period between doing the work and getting the award is now so long sometimes that many people don't make it. So I call them survival awards. I mean, we've had people in their late 80s get a Nobel Prize, and I think one in their 90s. And some of them are awkward in that they seem to be lifetime achievement awards. There isn't one thing in particular, but a constellation of them that in the sum make up, in the opinion of the committee, a body of work that deserves a Nobel Prize. It's interesting what happens to people's careers after. You'd think it would be a launch pad for more work. It isn't. It's a down thing. And why? Because they're a name now. They become a celebrity. They get invited to all kinds of stuff. And that bleeds time away from their work, and their productivity falls off, for the most part. With some exceptions, two I mentioned right here, Barry Sharpless, and I'll mention him later, and Arden Panaputian. They become public intellectuals. Why? What they know about is the subject they got the Nobel Prize for, not about religion or politics or anything else. But they become, it's not a Nobel Prize, but that kind of thing happened with Wilder Penfield. A famous person invited to make all kinds of comments about this and that, and for which, I mean, I think he was a marvelous person and made major contributions, but the stuff he was being asked to comment on, he was no better than you or I don't think. So, lessons learned. Well, I've drawn up a mention to Richard Feynman, the American Einstein, and he had he made the point, loud and clear, very, very clear, that without evidence, it doesn't matter what the theory is, who made it, what their name is, what their reputation is, if it doesn't, if the evidence isn't there, it's wrong. It's that simple. That's how science works. And you'll see that in play with Albert Einstein as I go through a little bit more here. And Why the delay in Nobel Prize? Well, sometimes, as I mentioned earlier, it takes several decades between the doing and the acknowledgement, and recognizing too that there's a stack of applicants or people lobbying for applicants, waiting in a line for a Nobel Prize, and their names keep coming up every year. I'm sure it happens in literature, and it certainly happens in science. Sometimes the Nobel process and the Nobel laureates make mistakes, and not many, fortunately, inside. Chair right up here.
Oh, good, yeah, cool.
Yeah.
Okay.
Yeah, and but also the laureates make mistakes. I mean, I'll tell you, a hand-footed example that I mentioned just a few minutes ago was Einstein. I mean, he was tripped up by his own masterpiece. How often does that happen? He was tripped up by general relativity because he didn't realize what the implications of general relativity were. And when others started to look at the equations, they realized, holy mackerel, the universe is expanding. He was really opposed to that. And he thought, well, just like Isaac Newton, he had a strong view that there was no beginning or ending to the universe, and it was a static affair. And so he told Lemaitre, he said, well, you know, your mathematics may be good, but your physics is terrible. Same thing with Schwarzschild, who came along, and he said, well, you know, if the mass is large enough, it'll collapse space-time around it. Well, what is that? It became a black hole. People realized that that was the genesis of the black hole, and also the kind of thing that created a singularity, a very tiny ball of intense energy that became the universe. So the Big Bang. So he could have had a Nobel Prize for at least taking the first step toward an expanding universe and the possibility of a Big Bang. But he didn't. And there were some other things that he did too. So even the bright make mistakes. That's important too. Now, I have favorites. And this is the 8th year, and so these would be my pick. I was really, the first year, I think, was 2017, and it was gravitational waves. Well, that was a prediction of Einstein. That was it was one of the throwaways. Meaning he said, well, you know, if two large bodies, massive enough, collide with one another, they're going to ripple space-time, just like throwing a stone in water. And Einstein's really reflecting on that said, well, you'll never be able to detect the waves would be too small. Well, it was true at the time. But then sensitive enough devices were developed, and they were detected. In 2015, 2 years later, the Nobel organization gave a Nobel Prize for it. But that was one of his creations, gravitational waves. Frances Arnold, I pick out in 2018, not only because of the extraordinary nature of her, which I've alluded to before, but you know, proteins, they consist of 20 amino acids. Can you imagine the permutations and combinations of the order of those amino acids, particularly if you're changing the number of them? I mean, it's almost impossible to come up with some rational way of developing a protein. And she came up with this seemingly crazy idea, well, let's let evolution do it. I'll introduce mutations into some proteins that I'm trying to develop as enzymes, and then they can battle it out, and I'll find out which one of them is the most productive, and I'll go through several cycles of that. And some of these enzymes became hundreds of times much more effective, using evolution to do it. I mention this because some of the developers of AI have used the same principle, a similar principle, meaning self-learning devices that will actually sort out among within the program, work out which solutions are better than others. So it's kind of a form of biological evolution in AI. And so that's a very important principle. I mean, that's why AI is so powerful. It's one of the big reasons, because it literally has a mind of its own. And it does evolve, maybe not quite the way we think about it with biology. Black holes, well, they didn't award a Nobel Prize for anything to do with a black hole until 2020, because no one had seen a black hole. Then a year before they did. And then all of a sudden, we have a Nobel Prize awarded by the Nobel Committee. And one of the, Roger Penrose, the mathematician in the group, why did he get his share? Well, he's a mathematician, for showing that general relativity was right. So long after Einstein was dead, gone, here's the only time the Nobel Committee actually acknowledged the correctness of general relativity. It's an amazing backward way of doing so. And we saw that play out 2 years ago with one of my favorites here, the posthumous end to the great Niels Bohr-Albert Einstein debate. I mean, this went on. These two men liked one another, they enjoyed one another's company, and they argued like crazy about whether quantum mechanics actually made sense. And Einstein's final challenge to Niels Bohr was, remember I've illustrated this before, quantum mechanics allows for two particles to be entangled, meaning they do the same thing at enormous distances. Einstein said that's nonsense. It can't possibly happen that way. Well, it was actually tested 30 years later, experimentally. And it does happen. And quantum computers depend on entanglement. So he was wrong on that again. And this year, I've added to the list what people were talking about today. John Jumper, especially, Denise Hassabis, and Google. I'm including Google in here because this would not have happened without a giant like Google getting directly involved, not just in the funding, but in picking and choosing the right people and encouraging them and boosting them along, and then handing it all out for free to the public, to meaning to scientists. So it's really, they didn't get named, but they could have been. Maybe should have been. By the way, there is a lovely book I'm enjoying right now, the quantum drama, from the Bohr-Einstein debate to the riddle of entanglement. It's a wonderful book. It's a wonderful story about two men that I have great respect for, and they must have been formidable friends, and but also antagonists when it came to quantum mechanics. But they really liked one another. Well, you don't need to see that again, but well, there's Frances Arnold, and I already mentioned what she did. She brought evolutionary principles to bear on the selection of a better enzyme. That was an extraordinary thing. And then here's another favorite, Jennifer Doudna and Manuel Carpentier in 2020. I thought this was the peak of stuff in 2020. You know what? The world is moving fast. Their technology that they developed, it's not passé, but they moved along. They made it better. But they've also moved away from gene editing and now say, well, listen, why go after the gene when that could we can make errors with that. Why not go after the messenger RNA? After all, genes create with the template for messenger RNA, create the template for a protein. So you're really just intercepting, or you have three targets. You can go after the proteins, you can go after messenger RNA, or you can go after the genes. Well, they went after the genes. And certainly there's a lot of good work going on. But I'll tell you, look at the direction of where pharmaceutical companies are going these days, where they're see the greatest development, is going after the messenger between the gene and the protein. So, good step. Well, I don't need to talk about gene editing here, but, and then I put this one up. By the way, you notice it's Swedish. One thing I've noticed is that any message in Swedish is shorter than English is shorter than French. And they seem to get to the point quicker. But my hero is Carolyn Bertozzi, very sharpless, really quite an extraordinary person. This was his second Nobel Prize. And he certainly deserved it for his hard work and vision. But Carolyn Bertozzi really stood out. I don't know whether several years or several months after that, her Nobel Prize award, I think it was on CNN, there was a quorum, a roundtable discussion, and they were all laureates of that year, but also a couple of years before. She was the grown-up in the room. She knew how to work with people. She knew how to lead and encourage people and bring them along, and she was quite a force. Is still, and a very imaginative person. So she not only knew how to run a very high quality organization, but she had good ideas herself. But she encouraged them in others. I mean, that's the whole thing with Niels Bohr, wasn't it? I mean, how do you know where the ideas came from in Copenhagen? Was it Niels Bohr or Heisenberg or any of 10 other people that were traveling in and out of the place and exchanging ideas? That's how her lab, that's what she encouraged, and hopefully is still doing it. And with the thing, I don't know how many of you remember, but this is a kind of a cruder, and I'll think of this, I shouldn't refer to anything she ever touched as crude, but this is only 2 years ago. And the idea here was, well, if you want to build better proteins, well, let's build them like a mechanical set from pieces of proteins and then clip them together, literally clip them together. Sounds like a good idea. It certainly worked for them, but it's a long way from where we're going with AlphaFold 2, which is today, and that's only 2 years. And so that was click chemistry, and I'm not going to get into her method here at all, but it was very good. The other thing, one of the pair I really enjoyed, especially Artem Patapoutian, fellow on the right, because this is a really classical neurophysiology. This is working out how we feel things on the surface of our skin, touch, pressure, temperature changes, that kind of thing, right down to the receptor, molecular, protein level, and the genes responsible. I mean, they were very elegant studies, very elegant. And so they studied the kinds of things, how do I feel texture touching this table, or this top here? And with the sensitivity, that exquisite sensitivity that we have. So I really like their work. And here's one of these receptors. This is the protein. This is a protein year for the Nobel. So, and this is a very large protein, one of the very, very sensitive mechanoreceptors, touch receptors, and it's shaped like a screw on a boat, right? The motor. Here are the blades. Here, it doesn't take much imagination because I can walk around here, but if you kind of push in on here, that these blades might expand, and what they do is open the pore, and ions go through the pore or whatever it's selected for. I mean, it's right down to the molecular level. These are very complex proteins involving thousands and thousands of amino acids. It's really quite extraordinary. And so I thought, wow, gene to that. That's pretty impressive and a very worthy Nobel Prize. And then last year, who could not have a lot of regard for Katalin Karikó and Drew Weissman, especially Katalin, because here's a woman who these were the parents of the mRNA vaccines, right? Which we are indebted to them for, because they provided the background work for the technology that was on the shelf that we could then use. Thank goodness. But she had a really tough time in her career. There were times when there was no funding for her graduate students. There was even a time when she had no salary. She couldn't get her papers published, probably because she was female. And she didn't hold grudges. So really quite an extraordinary person. And that came out in the interview, which I used at that year, not in the Nobel thing, but in the Lasker Prize. Lasker Prizes, by the way, precede the Nobel Prize, often within 1 or 2 years, and did with them. So, well, here we are. We're getting to the thing here. My job was made easy because I knew this was coming up. Why did I know it was coming up? Because last year, the Lasker Prize, the Lasker organization, awarded a prize to Denise Hassabis, so the two principal people here, and John Jumper of Google, DeepMind, for AlphaFold. So that's last year, 2013. 2 years after its explosive entry into the marketplace, and the science place anyway, for people to see. Quite the characters. I'm going to look at this. John Jumper, his PhD was 2017. That's 8 years ago. He's gone from a PhD to a Nobel Prize. That tells you the hotbed he was living in, but also his own capabilities. And it's really quite extraordinary. Hassabis is kind of earlier by a decade, and a decade before that, David Baker. David Baker was based in a university, the University of Washington in Seattle, and did not have a direct connection with Google. He got half the prize, quarter and a quarter here. So, well, to go on here, we have to say something about proteins, right? These days, I mean, one of the reasons why a chemistry and a physics prize was awarded for this kind of stuff is that they were working. I mean, we ordinarily think even 3 or 4 years ago that proteins are something we eat, something that we're made of. We think of it as biological. But at its most fundamental level, it is not. It is made of atoms. So we're talking about atomic physics. We're talking about quantum physics here, and compounds, simple compounds being formed, say, and that really dictates the shape of a molecule. Think about water, H2, two H's, two hydrogen atoms, and one oxygen atom. It's a very polar molecule because of those principles, underlying principles. So, so the two hydrogens are kind of are sitting up here, and the oxygen down here. The two hydrogens are positive, and they're loading their electrons to oxygen, which requires a negativity. So it's a polar molecule. That's what makes it such a good solvent, a universal solvent. Do you follow what I'm saying? It's that kind of level that these people were working, at least certainly included in their thinking. So that is really stunning to be working at the atomic level and working out the shape and function of a protein. That's amazing. And it's all happened in a relatively short period of time. So basic physics, my father would be very proud that I knew anything about this kind of thing because he was a chemical engineer. Natural selection, well, I'm going to say some more about that later because, yeah, I'll say about it. Anyway. Proteins are strings of amino acids, a linear string of amino acids. And if you could kind of strung it out, it really, no matter what the order, it really doesn't have any function. Where does it get its function from? Because it turns and twists on itself and forms these three-dimensional shapes, and it brings, let's say the one string is this long, and by the time you fold it up, you've got a little ball like this. So it brings amino acids that wouldn't normally be together in the neighborhood together. Do you follow what I'm saying? It's like mixing and matching, or at least mixing anyway, neighborhoods or homes on a street, juggling them. So homes might have been 180 Gage Street, 181 Gage Street, 182, then you juggle that. And you now have a three-dimensional shape. That's where the power of the proteins comes from, because depending on the shape, it can expose certain reactive groups at the atomic level, responsible for the action of the protein, but it could hide others. So it's really a fascinating process to create a three-dimensional model. So people knew what the order, or at least for many proteins, they knew what the order of the amino acids was. But translating that into a shape and therefore a function, that's a whole other game. And that's what took so long. So I took this right from the Nobel Committee because there we are, the 20 amino acids that go into proteins. And then the change with the twisting and turning of that, and you get this kind of many-folded this is in two planes here, but three planes. And exposing some active groups in here, cloaking others over here. That's the critical thing to catch in all of this. And so it was very difficult solving this. Now, I think I've already said all of this stuff, so I'm not going to repeat it. Well, a little bit of history here. This Christian Anfinsen, working at the NIH, showed that if you took a protein and it was all kind of folded up in its natural state, and you and stretched it out, it would pop right back if you let it go into exactly that same shape that it had before. It has a memory, and it will return to that shape, and therefore that function. And he concluded very early in the game, it actually led to a Nobel Prize, concluded that the amino acid sequence encodes the shape and therefore the function. So that's the basis of almost everything I'm going to say here that's important. So that's 1972. So we're talking about, half a century ago, or a little bit less than that. But here's what the problem, the progress is really extraordinarily slow in trying to figure out how do you translate the sequence of amino acids into a shape. Well, one way of going about a problem like this is turn it into a game. So here was the game. A group of people got together, led by this fellow and colleagues, and they ran every 2 years a competition. And the committee would, there were ways, which I'll mention in just a second, of figuring out what the three-dimensional shape of proteins was. And they would have a group of them all figured out, but they wouldn't tell the contestants. All they told them was the sequence of the amino acids, and then they could come with whatever programs they had and have a go at it and see what success they had in matching what it should be. Sounds good? A long time went by before any progress was made. And, oh, by the way, solving the riddle, here's how the classical people kind of figure out the shape, X-ray, crystallography, and right down to cryo. The important thing, these methods they're very precise, they're also very expensive, they take a long time, so we needed a better way. That's why we got to AI in this. So, and here are, the very slow progress here, starting with that group, the evolution of the number of experimentally determined protein structures using those tools that I just listed. It's a very slow thing. I mean, they started using those tools back in the mid-1990s. And, by the time, 2019, 20, using those tools, they got up to something less than 200,000 proteins. Pretty impressive. But given that there are billions and billions of proteins, it's not a lot of progress. And here it comes. Slow progress, then AI and self-learning. Well, let me just go back here because this is the enter Google DeepMind. Google's a big, big company. They've got a lot of heft, they have a lot of expertise, they know a lot about computers, machine learning, the kind of stuff that we talked about last week. And they know how to solve stuff in small groups. Small groups of people tasked with a particular problem, and you give them the resources and say, go to it. And if you got the right people, if you chose the right people, there's a good chance it'll work. And it did. So back in 2018, that's not long ago, 6 years ago. And what's the big difference? In contrast to traditional AI approaches that relied on preconceived logic, they employed machine learning systems, which you might remember from last week, that look for and find patterns for themselves in the data that the investigators couldn't come up with. That's the key. AI on its own is able to figure out a solution and even develop the kind of algorithms that would produce those solutions. That's extraordinary. That's the big advantage. And when they applied the first version of this in 2018 to that competitive group, wow, they just beat the opposition. I mean, no doubt who won that year. But to their everlasting, this is probably one of the most important sentences here. This could have been the lap of triumph here, but the group that had so succeeded and produced such a triumph here, a 50% improvement over the best 2 years before, what they did is they said, that's not good enough. They ditched the whole damn thing. They said, okay, we're not just going to try and fix the software that we've got. We want accuracy to the angstrom atomic level. We want a precision that's as good or better than those machines that you've got out there, crystallography, etc., analyzing that stuff. That's our target. So the goal was high, and Google supported them, and so I think that's the key to this whole business. The Google support and John Jumper. And so look, this is what happened. So these are the success of these competitions. I mean, it's kind of up and down, the success rate, but not very impressive. Then that kind of early version of AlphaFold won. Well, certainly a lot better than the others, and that's what they ditched. And they started from scratch with AlphaFold 2. And it was just very, very impressive what they did. A very gutsy thing to do. And you see the same kind of thing here. I mean, this is the clunky way of doing it with crystallography and the other techniques here. Yes, they're accumulating proteins and sorting them out. Then AlphaFold enters. There's the first version. And this, the little break in the thing, it broke it wide open. And we're talking about millions of proteins that have now been decoded, and would you believe it, in minutes. That's impressive. That's very impressive. And the Nobel Committee didn't mess around. They got on this one and awarded it. So I think I've already said that stuff. So this is what they wanted: predictions that were accurate to atomic precision, etc. So, but getting to that, I wasn't quite sure with this group how much to say about this, but I am going to say about it. I am going to mention this because this was their process. And how did they actually go about this? And so I'll just mention they forced their AlphaFold 2 model to become much more efficient. And then I mentioned last week, if you run the program again and again, and it's like facial recognition. The first run through, you see little differences in, colors, and there's certainly no definition there. You have no idea it's a head, never mind a face. But through running the damn thing through again and again and again, it's learning and then incorporating those new ideas into the next version and improving on that. It's called an iterative process, meaning running again and again through the program. But the program's also learning and changing at the same time, compounding the benefit. So it's really quite extraordinary, the process. It's almost human-like in how it's learning. It's like us doing homework and going back over till we get it right. Or we're lucky enough to have a teacher that just gives us the right insight at the right time. And click, we've got it. They also dumped any old ideas. And the other thing that they did is said, okay, we're not starting from A to Z here, or at least we're not going to solve this thing from A to Z, beginning with A, B, C, D, E, F, and then solve the middle of the alphabet and assume that the first part was right. Every time, the whole thing was recycled. No assumptions were made going through each cycle that the previous one had it right anywhere. We don't do that often as humans. And they also, obviously, they fed all, if we had, they had 200,000 in their database, proteins that they knew. And they figured, well, listen, if they there must be some rules, underlying rules here that we can discern from this database. So they fed it with that database. But that was only part of what they did. And they also integrated genetic information in this, in the sense that as we evolve, we've certainly, species have changed, but so have our proteins. A little bit at a time. Our hemoglobin is not the same as the hemoglobin of a rhesus monkey. Not quite. And so nature's figured out some variability in all of this, so they included that genetic information in the thing. And but here I think is a real key. No single element was decisive on its own. In other words, no one person on the committee was in charge, had overall control. That was really important to getting it right. So this is a group thing in these algorithms here. We're talking about a very democratic process at the molecular and atomic level. And so it was really quite extraordinary. And AlphaFold 2 was an enormous improvement over AlphaFold 1. Now, where did David Baker come in this? Why did he get half the prize and the other two have to split it? Well, David Baker, remember he was the instrument fellow at the University of Washington. He flipped things and he said, well, by the way, he developed a very similar program to AlphaFold 2, and both his group and the Google group provided that for free to the public, no charge. But he turned it around and he said, well, listen, this could be used to design entirely unique, one-of-a-kind proteins. We could maybe make little molecular motors or new receptors, or very much better enzymes tailored for a certain thing, or make certain absolutely novel pharmaceuticals that have never been seen before, using AlphaFold or his version of it. So that was very impressive. And I guess that's why I got half the money. And this is one of the early ones. I'm not quite sure what this one did, but this has never been seen before. Take me at my word here. And here's I'm going to go through all of these, but here's an interesting one. Developed in 2021, which makes a lot of sense. Nanoparticles are in the ball in the center here, and that's the yellow part. And the proteins he created proteins that imitated the influenza virus, but you couldn't catch it. But immunologically, it would be a target for the body. Do you follow what I'm saying? Yeah, so very clever idea. And so you can see where the pharmaceutical industry is quite interested in this stuff. Even a little tiny motor here, molecular motor. So I don't know where that's going, but the tool is there, and I think it's very impressive. And I'm not going to go through. Well, the pharmaceutical industries, you can understand, are very, very interested in this stuff. I mean, listen. What is it? 3 or 4 years ago, we knew about 200,000 proteins. Now we know about 200 million proteins. And in almost no time at all. Now, the next thing that caught my eye that nobody mentioned, but I'm going to bring up, Svante Pabo got a Nobel Prize in 2022, and he's one of my favorite fellows because he established the whole field of ancient DNA. And so Neanderthal DNA, pre-humans before that, and certainly early humans, and an incredible feat. But there's a problem with this, a built-in problem here. DNA is very fragile. You know, it's well preserved in dry, cold areas, but if there's any dampness or humidity, that kind of thing, the stuff is destroyed in no time. But you know what hangs around? Proteins. Proteins are far more durable. And I think the Paleolithic tool of the future is going to be proteins, not DNA. Because now you can move back millions of years, hundreds of millions of years, at least for some proteins, even 1 billion years plus. And you can actually, if you're talking about a creation story, we have a creation story soon to be written here in detail about some of the most important molecules in our body and when they began and how they evolved and in response to what. And so whereas Pubble could go back 600,000 years, this is going back by a factor of 100 times that, 1,000 times that. And that's really quite extraordinary. So you can be dated fairly quickly with a Nobel Prize here. I'm not saying the Karolinski Institute is going to fold and wrap up the program. Of course not. But I think, and this is what I kind of put on a slide here: DNA very fragile, proteins very, very durable, and the time scale here. So I think it's, now there's a video here, and I'm reluctant to play it because it's, Debbie set it up, but I think it's, what time is it? 49. I'm going to lay off the video a little bit because it goes on, it will go on well beyond our time. But let's open it up now. And some of you maybe have some questions. Any thoughts? Yes, Steve.
My mind kind of boggles when you say 200 million proteins.
Yes, 200
Now,
proteins.
They're given names?
No, they don't have individual names. We don't have a dictionary that large.
Well, that's what I was confused at. Who says there's 200 million.
Oh, because of the distinctive order of the amino acids in the protein.
So they have actual visual proof of this, then, do they?
Yes, they have physical chemical evidence of the order of the amino acids in these proteins, and much more importantly, they know how they folded. Because the function comes from the folding.
So they would have records then of 200 million proteins someplace stored?
They do. AlphaFold 2
Oh boy.
is incredibly fast. And they were so smart about this, releasing it to the public. Well, public, not us, although we could do it, I guess, but the science world. In no time at all, about 1 million labs kind of signed on. And we started to use the AlphaFold 2. So that has a huge amplifier effect, doesn't it? Because no one has any idea what all those people are going to use it as a tool. Was it just a look-see or a curiosity or what? Or is it real meat? But even if 1% of them have meat, or potential meat, investigative meat, then that's definitely worthwhile. It's also, one of the important things I hope has been driven home with this week and last week, is the steepness of the curve of development. That is extraordinary. I don't know of anything else in, at least certainly in biology and much of science that has had that explosive impact and multiplier effect. And it hasn't been because we were stuck for decades with, and I bridged that era. I had people working for me, writing programs to do stuff that we wanted to do in the lab. And we were very specific about what we wanted to do and look at. And the marching orders were very explicit. But this is different. This is very different. There you were writing every single piece of code to do a specific thing. You didn't want it to go rogue or do anything else or develop any independence. But machine language is independent. It's quite capable of, because of its very large database, and because it's free to incorporate useful algorithms in its database or anyone else's database that it can lay its hands on, or a better algorithm. Thinking back to harnessing evolution to make better enzymes. The principle's the same. And so we have software that has an independence that software never had before, and it's acquired it in the last 2 decades. Yes.
Pharmaceutical companies have to charge huge amount of money for any new vaccine or medication that comes along. With this now AlphaFold 2 being given for free, and you're saying the trajectory is going so quickly on research, does that mean that the cost should come down because they don't have to spend as much time researching, or it's not related?
In my experience with pharmaceutical companies, I don't think there's any altruism of that order.
So they won't pass along their savings of time and money and research?
We would hope so, but I don't see it. Maybe I'm shortchanging them, but I don't think so. And yeah. Oh, sorry.
Another question.
Yeah, anything else? Yeah.
My name is Steve Goodfriend. I'd like to pose a devil's advocate question that kind of challenges a couple of the assumption of genome to mRNA to protein. That relates to prions or prion diseases.
Prions.
Yeah.
Prions, yeah.
So there's a couple of possible assumptions that, I'd like your comment on. If somebody consumes contaminated meat, in the case of mad cow, and gets Creutzfeldt-Jakob disease, that implies that a foreign protein continues to propagate either itself or malforms my own proteins, possibly in absence of an mRNA that generates
It does.
a defective protein.
It bypasses the gene and mRNA.
So the assumption is, you just said that like all of the proteins are, the peptide sequences are dictated by the gene, by RNA, and then to the sequence of the gene that then folds on itself, the sequence of the protein that then folds on itself to create a shape. That model is pretty much thrown out by prion disease.
Yeah, a lot of neurodegenerative diseases consist of unfolded or misfolded proteins.
Yes, misfolded, unfunctional,
Misfolded
non-functional.
and unfolded
Yep.
proteins. And the prion part that you're referring to refers to their capacity to induce the same changes in normal proteins. And hence the sense that it's an infection, meaning it spreads in the nervous system. And in a way, it certainly does. Croup and bovine encephalopathy and CJ disease, they definitely spread, and they can be transmitted.
Right, so my point here, and you're saying
Yeah.
the same thing as I am, is that here's this non-functional protein,
Yeah.
a prion, that will influence the shape of another protein.
Its own protein that happens to have a normal shape.
Okay, so if I consume a single prion molecule from eating an infected cow, can that one single molecule migrate into my brain and then influence the shape of other native proteins in my brain, native functional proteins in my brain, to create the condition called Creutzfeldt-Jakob disease? So that in the kind of fundamental question that I'm throwing there,
Yeah, that's.
can a single misshapen protein molecule create a Creutzfeldt-Jakob syndrome? Because that implies that it's commandeering the whole protein synthesis and shaping mechanism of my brain.
Yeah, it certainly, when this whole business was brought up and the discovery was made in New Guinea, and people eating brains of people with this disease, there was a great reluctance. 2 Nobel Prizes been given for prions. And there was a great deal of opposition because people simply couldn't buy that this was an infectious agent. But the argument was, well, if it looks like one and it behaves like one, it is one. Even if it isn't a cell or a virus or a bacteria, it's still behaving like an infectious agent, meaning it's taking over, it's spreading. And in the nervous system, that often happens from, cells are connected to other nerve cells. So if cell A happens to have a mutant form or a prion form, that is transported down the length of the nerve fiber, across the synapse, into adjacent, up to then, normal nerve cells. And it's quite capable of in the proximity of what are apparently normal proteins, converting them or distorting them, usually to an unfolded state where the protein doesn't work.
So
it's very.
So if I could throw out one of your slides, said, and a Nobel Prize was awarded, that the Nobel Prize recipient unfolded these proteins to a single strand,
Yes,
arguably,
yeah.
and they spontaneously refolded to their functional shape.
Yeah, except in this case with prions. They're
So
not
that's where
they're not springing back.
so that's why I want to drill down. If that's not a function of the primary sequence
Yeah,
of
and-
peptides,
And furthermore, to the best of my knowledge, and I certainly could be wrong, I'll look this up when I leave, but I don't think anyone's actually established the structure of prions at the amino acid level.
well, Google should be on that.
Well, yeah, it is. I mean, it certainly is scary. I mean, I remember when I was at University Hospital in London, and there was a case on the floor, and in those days they, the cases were pretty infrequent, and people wanted a diagnosis. Well, how do you get that in those days? You biopsy, which meant a neurosurgeon went in, gloved, but unfortunately cut his glove. And boy, was he, and of course with a long incubation period of 5 years, 10 years, 15 years, how do you know? You don't know, because they might have died from something else beforehand. Or whatever, but boy, did that ever get people upset, and very understandably so. These days, they don't need to do a biopsy. Somebody doesn't have to expose themselves to go in and get tissue that way. But it's a scary business for sure. And now, some people argue that Alzheimer's disease, Parkinson's disease, frontotemporal degeneration are, they have prion-like qualities, meaning the abnormal, they're all fundamentally protein problems, and the proteins are abnormal. And the abnormal proteins are spreading in the brain, like prion diseases. But there's no evidence to date in those 3 cases, instances I mentioned, of it ever being transmitted to another human or to another species. So yes, maybe it is behaving like a prion and spreading without mRNA or gene, just protein to protein. And maybe so, but it's seemingly not catchable. But then mind you, if that happens in somebody in their 70s or 80s, they'll probably die long before they reach the time that they would ever manifest any symptoms. So there is one team in the United States, I didn't highlight it here today, but there's a married couple working on the team. And she has the form that produces insomnia, pathological insomnia, to the point where people simply can't sleep, and that's very destructive, as well as some other symptoms. And she has no symptoms now, so they're working breakneck to see if they can solve the problem before she develops the symptoms, which she will 100% get because of the disease. So, it's I'm glad you brought up the prion thing, because how does an affected protein, in a way, it's like a kind of a spoiled neighbor or whatever it is, going door to door, and how does he change each person along the street? How does that actually happen at a protein level? I don't know. but it certainly, one of the, I mean, this all sounds like a very rosy picture, doesn't it, about developing proteins that break that neck speed and with spin-offs and might be very helpful, certainly to the pharmaceutical industry and maybe to the human story. But it's also an intelligence that clearly exceeds ours in certain ways. Humans could not do any of this stuff. They were scuppered for 2 decades. Very bright people working on this stuff until machine language was incorporated and the database was large enough to learn on it and become clever. But it's that independence that it has, that's what's novel. That kind of takes paths. I mean, there have been many instances reported of a program kind of taking a different step and it's much more efficient. And the programmer standing back and saying, well, how the hell did it do that? And having trouble figuring out which algorithm or how the algorithms actually changed. But they do. I mean, as with us, we speak of mutations. Well, you could use a similar term applied in this case to computers, that every time it replicates doesn't mean it replicates exactly the same. And if it doesn't replicate exactly the same, its properties may actually change. And that's how we got to be where we are on Earth. But we're talking about a machine doing that on its own without our input. That's the extraordinary thing. In the first talk we had here, we didn't kind of get into this because we were really talking about more theoretical stuff in earlier days, 1980s and early 1990s. This week is very different because this is a very specific goal. How do you figure out the three-dimensional shape and probable function of proteins? Very specific. And weren't asking any other questions. And so the success is just from the graph. It's off the graph in 2021, 22. I haven't seen a graph since then, but I can quite imagine it's continued to spike, going up. So what does that do for a whole lot of things that we use computers for? Surveillance systems. So anyway, any other scary questions? Yeah.
Just to make one general comment.
Yeah.
I think we can expect one heck of a lot of development in this area. And we now have an AlphaFold 3. And as we use academic institutions and as companies use this to go after some of the really, really serious diseases we've got, and developing new enzymes that can correct old enzymes and new proteins that can correct old proteins and new drugs that will attack these very, very bad situations in terms of spreading of disease. We're going to see a lot of, maybe more Nobel Prizes too.
That's why Google released it. It's also why the Nobel are shining a light on this right now. Exactly that. Because there is an AlphaFold 3. And there'll be an AlphaFold 4 and versions beyond that. But corralling it, or whether we can corral it, can put Jack back in the box, Do we want to check back in the box? I don't know, but anyway, we can speculate all we want. Now, next week is more traditional. It's actually my favorite, not because it's traditional, but because the people involved are among the best. And I think I mentioned to you last week, next week's Nobel Prize, so far, there have been 8 Nobel Prizes awarded, or Nobel laureates, all about a 1 millimeter worm, working on a 1 millimeter worm. And why that? Because the systems are simple, they're complex enough to be analogous to ours, meaning conclusions you reach on that tiny little worm apply to us. But they're simple enough that it's much easier to figure out what's going on, because you can actually see every single cell and every single connection in that tiny little worm. And it created quite a community among those people working on it. And they held, I think, as much social as anything else, meetings on a regular basis to talk about their C. elegans. And elegans is what sounds like elegant. And and it's worthwhile because the caliber of the people involved is really quite extraordinary next week. So we'll see you next week.